Okay, this is a little mini presentation on the sort of things that we'll be seeing. First, I want to give you a teeny tiny overview of animal evolution and the broad evolutionary groupings. We're going to be seeing these in Paradise, so that makes it a bonus, and maybe you'll learn some things without even realizing it. Prior to going to Roatan, however, we have some stuff to accomplish in the Twin Cities, and we're doing this in an abbreviated fashion because of the New Year's holiday and all that. So our class time is going to be pretty compressed. That's why I'm asking you to view a couple of these short lecture slideshows, and hopefully it won't be too painful. We'll talk about logistics, but we'll also go over things like animal evolution or phylogenetics. We'll talk about who occupies coral reefs and how they're constructed, who the corals actually are. We'll go over some threats to coral reefs, including some kind of point source things that maybe you'll be able to detect in our travels. And we'll talk about reef biogeochemistry in a nutshell, not trying to scare anybody. The goal for our final Friday product is to have a testable hypothesis or a series of testable hypotheses in our groups that we'll address during our dives and snorkels at the Roatan Institute for Marine Sciences. The way we'll accomplish that is on Wednesday, our first day, we'll go over our course goals again. I am going to discuss animal evolution and taxonomy in a nutshell now, but you'll have a quiz on it on Wednesday. Fun. And we'll also go further into our discussion of threats to coral reefs. I am going to have a small presentation on coral reef ecology as well. You guys, though, on Wednesday will also be presenting to us in small groups about literature that I've assigned. So we'll I call that a lit smattering because it gives us an idea of what kind of work has been done on coral reefs, what might be feasible, what questions we could ask, etc. The idea is that then you would, that might trigger some interests and you guys could go on and find your own papers for Thursday and Friday discussion. On Thursday, we'll probably have a shorter day than Wednesday. We'll talk more about coral reef biogeochemistry, and you guys will present on papers you bring in. We'll be sorting into groups of probably 2, maybe some of you will want to work as individuals, and there might be the need for a 3-person group. But this will be to generate questions and come up with a testable hypothesis for your time in Honduras. And then on Friday, we'll have final group presentations on what your question is, what your hypothesis is, what you might need from the group in terms of help in the field, and we'll try to troubleshoot so that we're as ready as we can be given how crazy field research can be. But that will be, hopefully, will feel like at the end of the day, Friday, each group or individual has a hypothesis that they're ready to test underwater. This doesn't mean you're going to be testing your hypothesis or collecting data the entire time you're diving. We're going to have fun and we're going to absorb our surroundings as well, but you'll at least have something that you're hoping to come back with. Okay, so today let's talk a little bit about animal evolution. We'll talk about the characteristics of animals, what makes something an animal. We'll look at major evolutionary branch points, so how we sort animals, and talk a little bit about their basic development and broad classification. We'll use broad terms like cnidarians and arthropods, crustacea, etc. that we'll all want to be familiar with while we're down there. And you, of course, will be identifying a lot of different animals. Sometimes you see the term kingdom used to describe the animals that's within the domain Eukarya. However, kingdom is sometimes considered an outdated term, so you can just say clade Animalia if you want to be evolutionarily accurate. So I want you to think about, first, well, I want to mention that the animals showed up within the last billion years, probably. There's fossils of what we recognize as animals from over 700 million years ago. They may have evolved from an ancestor they shared with a protist, a choanoflagellate, or a thing that had this collar-like flagellum, single-celled organisms, though, that lived in colonies. So we refer to these as colonial choanoflagellate ancestors. You can see here the broad groupings of the animals and how they fall out. These aren't the broad groupings of the animals. These are the broad groupings of the eukaryotes. So the eukaryotic organisms are the protists, things that we really don't know what to do with taxonomically, the plants, fungi, and animals. So a question is, what makes something an animal? Okay, what separates animals from other organisms, for instance? So animals, for instance, are multicellular, but that's certainly not a hallmark of the animals because plants and fungi are multicellular as well. So think about that. What makes something an animal? They have a suite of characteristics that unifies them. They're motile, they can move around. They're multicellular. They're heterotrophic, which means they rely on other food sources. They can't generate their own food the way autotrophic organisms like plants and cyanobacteria can. They ingest nutrients. They store their carbohydrates as glycogen instead of other forms of carbohydrate storage, starch, for instance. They have characteristic nervous and muscle tissues and a development that is unique to the Animalia, and that's what I'll say more about. We recognize various levels of organization in animals. Protoplasmic is a weird term, and it applies to animal-like protists, but these aren't really animals. Animals like sponges, for instance, have a cellular level of organization. We get into tissue level of organization with the cnidarians, the jellies, and the corals, the anemones. And then we start seeing organs levels, tissue organs, with things like flatworms, the platyhelminths. We'll see some platyhelminths. And then organ systems we see in other animal phyla, like the chordates, like ourselves. We recognize two major evolutionary branches within the Animalia. The parazoans are those things that lack distinct tissues. Sometimes these are called the mesozoa. The eumetazoa have true tissues. Eumetazoa actually means true animals, but parazoans are true animals as well. So who are the parazoans? Who are the animals that lack distinct tissues? They have cells, they have the characteristic development of animals, they're heterotrophic. It's the sponges, okay? So parazoans or sponges you will encounter. These are all over marine habitats. There are also freshwater sponges within a group, the Spongillidae. You won't encounter those, obviously, on our trip to Honduras. So besides the split between the parazoa and the eumetazoa, there go the 10-year-old boys, there are other large evolutionary branch points. So here, moving left to right, number 2, we see another branch point whereby the eumetazoa can be divided into those things with radial symmetry and two tissues, two embryologically derived tissue types. These are referred to as the diploblasts. They are diploblastic. And then those organisms that have bilateral symmetry and are triploblastic. They have 3 embryonic tissue types. We also refer to these radially symmetrical organisms, or the diploblasts, as the radiata, and the triploblasts, the bilaterally symmetrical organisms, as the bilateria. So a word on developmental biology. When we talk about development, we're talking about a series of irreversible events from the zygote, the fusion of sperm and egg, to death. And as I mentioned, all animals have a characteristic development, at least for the first several divisions. Now, if you look at, for instance, mammals and turtles, we look a whole lot alike for a long time during our development. But for instance, mammals and sponges, it's only those first several divisions where we are clearly related. Okay, so we begin with fertilization, the fusion of sperm and egg that gives us a zygote. This is a single celled and it has the full complement of chromosomes that we'll have for the rest of our lives. We undergo early cell divisions that we refer to as cleavage. And cleavage gets us to this ball of cells that's referred to as the morula. Further divisions lead to a hollow ball called the blastula. The inside of the blastula is just a space. It's a body cavity called the blastocoel. In some organisms, that part of that blastula folds inward. You get an invagination or a pocket called a gastrula. This invagination or the forming of the gastrula is called gastrulation. And it's the gastrula in animals that leads to distinct tissue types. So the blastula is simply a hollow mass of cells, and it's the first tissue, first collection of similar cells. The blastoderm gives rise to other tissue types. And we refer to that hollow center, the blastocoel, as the first cavity. Gastrulation is where some of those surface cells of the blastula move inward, and that movement is accompanied by induction, whereby some of the cells are induced to become specific types of tissues. These induced cells differentiate, which basically means they acquire new characters through differential gene expression. So some genes are turned on or off depending on where the cells are located in the gastrula. That movement of cells inward, the little pocket that forms, is called the archenteron, and that is the future gut cavity for animals that undergo gastrulation. So we still have a blastocoel as well as an archenteron, and we start to see tissues differentiate into different types, endoderm and ectoderm, for example. Now, as some of these cells move inward, new embryonic tissues can also form. In the diploblasts, the animals that have two embryonic tissues, these are the endoderm and the ectoderm. And their fates are pretty much sealed at this point. What those cells will go on to become is determined once gastrulation has been accomplished. Some animals have three embryonic tissues. These are the triploblasts, and they have endo and ectoderm as well as mesoderm. Mesoderm is distinct in that it goes on to become muscle tissues and gonadal tissues. So the muscle tissue is key because triploblasts that have tissues derived from mesoderm are capable of more refined movements than the diploblasts. We also talk about organisms in terms of the symmetry they possess. Some organisms possess a spherical symmetry, and they're coming back in. Some are radial, and this includes the radiata, and some adult bilateria, like sea stars that you'll see. And then there's bilateral symmetry. Most animal phyla, including the chordates, vertebrates, the fish you'll see, etc., have bilateral symmetry. You can see here an example of how radial symmetry looks, where you have an oral and an aboral surface, versus bilateral symmetry, where the organism can be divided into a front and a back, or an anterior and posterior, and then a dorsal and a ventral, up and down, basically. And here's some examples of radially symmetrical organisms, like the sponge to the left, and bilaterally symmetrical organisms, like the nudibranch or the sea slug to the right. Now, some organisms we see have a true coelom, whereby the mesoderm is hollowed out to form an internal fluid-filled body cavity. So acoelomate organisms lack this, okay? The mesoderm basically just fills up the internal area between the endo and ectoderm, okay? So there's no actual coelom. We see this in the platyhelminths or the flatworms. Some pseudocoelomates have a coelom that is surrounded on one side by the endodermally derived tissues and on one side by mesodermally derived tissues. An example of a pseudocoelomate is a nematode, or a roundworm. True coelomates have a coelom that is completely surrounded by mesodermally derived tissue. An example would be annelids, arthropods, chordates. Those are all true coelomates. Pseudocoelomates, like roundworms or nematodes, the roundworm Ascaris lumbricoides is on the left, versus true coelomates on the right, you can see represented here. True coelomates include the cuttlefish, upper right, and the kitty cat, lower right. We also make a distinction between protostome coelomates and deuterostome coelomates. And this really has to do with what comes from the blastopore. So the blastopore is basically where the invagination in the gastrula occurs. If that becomes a mouth, then we talk about them as being protostome, mouth first coelomates. If that becomes an anus, we talk about them as being deuterostome, or mouth second coelomates. Protostome coelomates include arthropods, like the spider at the left. Deuterostome coelomates include the echinoderms, like the brittle star on the right, and all the chordates, like ourselves. There's other things that distinguish protostome coelomates from deuterostome coelomates. The way their cells divide in the early cell division stage, early divisions, the way the coelom forms, and also, like I just mentioned, the fate of the blastopore, whether it becomes a mouth or an anus. So we're going to see a bunch of animals from these different groups: protostome coelomates, deuterostome coelomates, pseudocoelomates, bilateral acoelomates. We're going to see some ctenophores, and we're definitely going to see a lot of cnidarians and sponges. We also can make a distinction about segmentation or metamerism. Some organisms have a repetition of similar body segments. Sometimes these are called metameres or somites along the longitudinal axis of their body. Which critters have this type of segmentation? Think about it. And perhaps you came up with annelids, like earthworms, arthropods, you'll see, for instance, lobsters, and chordates, like fish. Segmentation is pretty obvious in scaly fish. So I'm going to ask you guys on a quiz, for instance, which of these will we see? And give me some examples. Will we see poriferans? Will we see cnidarians? Ctenophores? Platyhelminths or flatworms? Rotifers I didn't mention. They're another example of possibly a pseudocoelomate, although I'm pretty sure that their classification is changing. Nematodes, the roundworms. Mollusks, like clams, snails, octopuses. Arthropods, will we see them? What kind of arthropods will we see? Annelids, the segmented worms, echinoderms, and chordates, which we can break down into the urochordates, the vertebrates, the actinopterygii, the amphibians, the reptiles, the birds, the mammals. Who will we see? And that is our broad overview of animal taxonomy and a little teeny bit on animal evolution. There will be a quiz.